All right, any questions from yesterday? Remember, we're going to try not to get behind at all, just because this is all going to kind of snowball. Again, the exam will be a week from next Monday over special senses and hematology. Same general format as what you did last week. Tentatively, so don't take this to the bank, but those of you who are curious, more than likely I will be teaching the ANP3 that's on Tuesdays and Thursdays in the afternoon. So, and then if unfortunately you don't pass this class, I think I'm teaching the earlier ANP2, but fingers crossed. Plan for not passing the class. So, but don't, there's just a couple more things that have to be tweaked, but that's the general plan. I will probably do an eye dissection next week. It'll be probably Monday or Tuesday. Tuesday more than likely. And then that's probably when we'll also start hematology. So just to give you a heads up on that as well. All right, so we talked about special senses. Remember, we divided them up. Chemical. Chemical is where we actually have, based off of the name, a chemical coming in contact with the receptor, and those are taste and smell. Remember, they have to be in solution. For the mouth, the solution is our saliva. For smell, that has to be in a gaseous phase, volatile. Again, that's what volatile means, is in a gaseous phase. That's the only way that the odorants are able to get into the air and then go up into our schnoz, up into the olfactory epithelium, which is in the superior portion of the nasal cavity. So I went through all of the anatomy from that standpoint and the physiology, and that takes us to the eyes. Okay? And again, as I stated yesterday, the eyes and the ears, lots of anatomy, lots of physiology. So again, that's why I wouldn't get too lost. Now, for those people, I've had some students every now and then that when they meet with me at Starbucks or something, they show me that they've basically drawn, you know, like this picture or, you know, some of these larger pictures, and they, you know, they've labeled it correctly. And I mean, the thing is huge. But since they spent so much time on it, they didn't get to the ear. So if you're an artsy-fartsy person, keep in mind that you probably don't want to spend a lot of time going into depth on making things look pretty. I would probably spend the time on trying to learn the anatomy. Okay? Now, the eyeballs. So the eye itself is the thing that's inside the orbit. All right? And keep in mind that when we see an eyeball, we're really only seeing about 1/6 of the eyeball, to give you an idea of its size. I mean, it's only about an inch, but the rest of it, the 5/6, is actually buried inside the orbit itself. And it's not a completely round structure. Your book kind of alludes to the fact that it potentially is, but if you were to take a football, poorly drawn football, but a football nonetheless, and pushed it a little bit more. So there is a pull on both ends. There is a distinct pull, an anterior and a posterior pull. So it's not a ball, but it's not the football either. So think about just taking the ends and pushing them together, and that's the general structure that is the eyeball. Now, for the eye to function, for protection, for movement, we have accessory structures. And we're going to go over the accessory structures first before we start tackling the actual globe, which is another term that we often use for the eyeball. Now, the structures, some of them, we really don't know what they do. Okay, the eyebrow is the beginning part of that. So this thing up here supposedly keeps sweat from rolling down into your eyes. Okay? Also supposedly protects the eye. Well, unless you have something that's completely untrimmed, it's not going to protect anything from flying into your eye. And those of you who work out on a regular basis know this really doesn't stop sweat from getting into your eyes. Right? So the eyebrow is really one of those functions that we're not quite sure what its overall purpose is. But as we start to move down closer towards the orbit itself, we have the eyelids. Now you should probably know the term for the eyelids, which is a palpebrae. That's the general term. You have a superior palpebra for a singular and an inferior. So that's just the name for the upper eyelid and the lower eyelid. they kind of form the opening into viewing the eyeball. So when the superior palpebra, the upper eyelid, the superior eyelid, is actually moved, and that's the movable one. When you think about it, when you put your hands on your eyelids, it's the upper one that moves. The upper one goes up and down. It's really not the lower one. I mean, you can move it this way, but it's not really moving upward to close. The top one's moving down to close, okay? When it's open, we have a thing called a palpebral fissure. So that's basically, if I can see your eyeball, then you have a palpebral fissure. If you close your eyelid, you can't see it, so the palpebral fissure is gone. Okay? So it's just the space between the top and bottom. And again, that's going to be gone if you close your eyelid. Now, as we look at it, it also kind of comes to a point over here. This is referred to as a lateral commissure. Sometimes if you have different textbooks, it'll be a lateral palpebral commissure, but lateral commissure is perfectly fine. And obviously, if I have a lateral commissure, I have a medial commissure. Okay? Right towards the medial commissure, we have this thing called the lacrimal caruncle, which is a fleshy part right up here. This produces a mucousy substance. Most of you, once you go to bed at night and that mucousy substance sits here, it dries out. This is what you all probably know as eye boogers that you kind of hope that you get out of your face before you go get your coffee at Starbucks in the morning. Your book says that this is the Sandman's eye sand. Eye boogers, I think that's better. Some of the changes or some of the modifications in this area, so individuals that are of Asian descent, they have what's referred to as an epicanthic fold. So it's a vertical fold that kind of covers this area on the medial component of the eyelid itself. And just since we're looking at it, we'll kind of go over some of the rest of them. Some of this you're going to get on multiple slides. So we have the white part that you can see is referred to as the sclera. The sclera is actually going to be also in the back. The sclera is going to be a layer of the eye, okay? As you can see it from this component here. We have the colored part of the eye, which is the iris. And then we have the entrance into the eye itself. This is how we see. So light is going to go through this black hole right here, okay? And the receptors for vision are on the posterior wall of the eye. The eyelashes, so when we look at the eyelids, I'll show you this picture here, it's a little bit better to represent it. So the eyelid itself is all attached to this fibrous connective tissue right here. It's referred to as a tarsal plate, okay? Now, in your text, you're going to see the term meibomian, okay? So wherever you see tarsal, you can also use the term meibomian. They are one and the same. Now, it is a point of attachment for muscles. So we have one of the primary muscles that controls just the eyelid itself. So I can blink without squinting. Remember, if I squint, that's predominantly the orbicularis oculi, the muscle that surrounds the whole eye. So to close your eye, yeah, I can squint and that closes my eye. But there's a more efficient, less energy process, which is I'm going to use the levator palpebris superioris muscle, which is in this area right here that's going to come down and attach to the tarsal plate, connective tissue, again also referred to as the meibomian plate, and that's just going to allow you to just move the eyelid itself. But again, we also have that orbicularis oculi that goes around the whole entire eye itself, and that is a point of attachment again to the tarsal plate, the meibomian plate. And then we have these structures down here, which really do have a mechanism, okay? Those are your eyelashes. Your eyelashes are protected from the standpoint that if you were to run your fingers over them, if somebody else does this to you and you're not really conscious of it, because you can, especially people who have contact lenses, they can sit there and stick fingers in their eyeballs and it doesn't seem to bother them. But for most of us, when you start touching the eyelashes, it causes a reflex and a closing of the eye so that nothing actually gets into and touches the eyeball itself. Doesn't always work. Bugs can fly in there a lot quicker. Pollen, not pollen, particulate matter can get in there, dust. So it's not the best thing. There are glands that are present at the bottom here, at the base, and it basically allows for them to be coated with the same kind of material that coats our hair, so that the hair can have individual strands as opposed to being clumped together. There's also a gland that's associated with the tarsal plate or the meibomian plate, and it's called the tarsal gland or the meibomian gland. Now, any gland can become kind of inflamed for a variety of reasons. So when the meibomian gland gets inflamed, it's called a chalazion. When the glands at the bottom of each of the eyelashes can get inflamed, it's referred to as a stye. And I will tell you, for the most part, if you go see your healthcare provider, unless they are an ophthalmologist, more than likely, if you have a bump in that area along your lid, they're probably going to call it a stye. Okay? The treatment for it is, they're basically just going to allow it to go down unless it's causing some sort of problem with closure of the eye, and then they could potentially drain it. All right. Any questions on that so far? Now we're going to talk about some of the protective mechanisms of the eyes. And we have a specialized epithelial tissue, and it's referred to as a conjunctiva. Conjunctiva actually starts over on the inside of the palpebra, both on the superior and inferior. So on the inside of your eyelids up there, you have this process of specialized epithelial tissue that's going to come down, and that's referred to as the palpebral conjunctiva. Now it's going to double over on itself, both at the top and bottom and on the sides, and then it's going to cover just the white part of the eye that you can see. Remember, I told you that that white part actually goes all the way back. So the conjunctiva only covers the white part that is visible. And it's going to go all the way up to a specialized structure called the cornea. So the cornea is not covered by the conjunctiva. Now, the part that's covering the eyeball is called the bulbar conjunctiva, sometimes the ocular conjunctiva. So the sheet, the epithelial sheet as a whole, is called the conjunctiva. Then we're going to divide it into two parts. The part that's on the eyelid, the palpebral conjunctiva, and the part that doubles over and then lines the eyeball is called the bulbar conjunctiva. Now, because it goes over, it bends over on itself, it creates conjunctival sac. So those of you who have ever had to administer ocular medication to a patient, you can do it one of two ways. You can do it the really unpleasant way, which is where the patient lays back, and if you're the patient, it's kind of like having a gag reflex in your eye. You kind of move your eye all the way up into the orbit because you just don't really. So you lay there and they put the drops in. One of the easier ways to distribute medication, not every medication, ocular medication could be done this way, is you just simply pull the lower eyelid out and you drop it in the sac. And therefore it's tolerated better by patients, and then when they close their eyes and move their eyeball around, it helps distribute the medication over the surface of the eye. Now, because this can be somewhat transparent, there can be an inflammation that shows up underneath. So you can have what's referred to as a conjunctivitis, where this part becomes inflamed, but it also allows for underlying blood vessels to become far more visible. Most people know the term pink eye as opposed to conjunctivitis. Pink eye can be caused by viruses, caused by bacteria. If you've had a child and who's had it, more than likely get a phone call from the school and your kid has to go home because conjunctivitis is extremely contagious. And we get very lax as far as, because without getting into the nitty-gritty of the whole pathophysiology of the disease, most of the time we say that contagion lasts about 24 hours. And then after that, you're good to go. But there are some that do last longer than that. So usually that's where the physician or your healthcare provider is going to give you a little bit more direction on when that eyeball is less contagious. Any questions on that? All right, so still on the accessory structures. We haven't left that yet. We have a structure up here in the superior lateral component. It's referred to as a lacrimal gland. And this is essentially what produces our tears. It also has a chemical in there called lysozyme. And lysozyme is in almost every body fluid. So whenever you're taking an exam, it's like, well, what fluids or what is the component of this? Probably 99% of the time, say lysozyme, they're going to get some credit in there. And it's basically a naturally occurring antimicrobial. Okay? And it's produced up here. We have these ducts that come down. And so your tears, or the water that's present in here, is going to wash over the eye, especially as you blink. You'll see this for patients that are in intensive care units. We like to tape their eyes shut, that people in a coma, because if their eyes stay open, then their eyes tend to dry out really well. So we like to keep their eyes closed so that that doesn't occur. And then as you blink, it just naturally washes over it. Now keep in mind, as I've stated before, if I'm constantly producing something, a fluid, I need to do what? Get rid of it. If I don't get rid of it, it's going to build up somewhere. Here, it would just, if there wasn't a pathway, it would build up in your eye, and you would naturally have overflow. So we have these two little holes over here. They're referred to as the puncta for plural, punctum for the singular, and it's the lacrimal punctum. And you have a superior and an inferior. And they lead to these little structures over here, which is referred to as a lacrimal canaliculus. Both of them would be a lacrimal canaliculi. They're going to come together into this structure over here, which is the nasolacrimal sac. Okay? Now here, it's going to then drain down into the duct, and the duct drains down into the inferior meatus. Bless you. Remember I talked about the cells that stick out, bless you. And then you've got these little recessed areas, it's referred to as a meatus. So you've got this inferior meatus, and that's where your nasolacrimal duct drains into. This is the reason why your nose runs when you cry. Okay? I mean, part of your nose runs is because of the parasympathetic system. You have an increase in that mucus production, but you also have the tears coming down through here. So if you have an issue with blockage, so we have the ciliary body. The ciliary body was composed of the ciliary process, the ciliary muscle, and then from the ciliary process we had the zonules, or the suspensory ligaments. And then we move up to the iris. Now again, right in the middle, we have that pupil. Now the pupil is going to be affected essentially by the two muscles that are present there. Now these are not the extrinsic muscles. These are intrinsic muscles. Okay, meaning that you don't really have a lot of control over them. These are basically controlled by the autonomic nervous system. Sympathetic nervous system, parasympathetic nervous system. So we have the sphincter papillae, sometimes referred to as the constrictor muscle. So when it tightens up, it decreases the size of the pupil. I let less light in. Now, when our sympathetic nervous system, we have a dilator pupil. I'm not even going to talk it. Papillae, or it's sometimes referred to as a radial, because what happens is it's going to get pulled out from that standpoint. It's also referred to as dilator from that standpoint, but you can see them right here. So they're going to pull, and when they pull, they make the pupil bigger. Now, since there's only two options, a good way to remember this is just to remember your fight or flight mechanism. Because if you remember your fight or flight mechanism, which is our sympathetic nervous system, our pupils are going to get big. Why? I want more light to come in. I want to see the dinosaur chasing me. So when more light comes in, I have a little bit better visual acuity. Okay, as opposed to parasympathetic, which my sphincter papillae are going to contract and let less light in. So one of the things that we'll do with the dissection is we'll also look at your own eyeball. So you'll take a pin light, and whenever you do check a person's pin light, you always want to kind of cover, and you always want to come from this side, because you don't want the light to go over here. Okay, because we're examining each eye separately. So a lot of times we get a little bit lazy when we're evaluating eyes during emergency, so we just kind of close the person's eye, their eyelid, and then lift it up. And when they lift it up, more light comes in, and you can kind of get a rough idea about what's happening. What we hope to happen is that this will occur. So if I shine a light in there, I'm hoping that your pupils will actually constrict. Why? It's a protective mechanism. I'm letting less light into the inside of the eye. So that's when you hear people say that when we do evaluation, pupils equally reactive, round, so we look at how the pupil looks, is there any other inconsistencies in it, to light. And we're going to talk about this last one in a minute. And accommodation, which means the pupil's going to change. There's going to be some structure in there as the light goes in. But you'll hear that someone says that they're pearly. Pupils are equally round and reactive to light. Sometimes they leave the A off and they'll just say pupils are pearl, because that's all they really care about. Pupils are reactive to light. We hope that both sides do it. So again, if I go in to see a patient and I raise their eye up and their eye stays dilated, and I flash a light in there and their eyes stay dilated, it's not a good sign. Now, it could mean that the patient just has big pupils, and that's not necessarily an uncommon thing. Or the pupils can be genetically unequal. So they sometimes will look at mom and dad and go, oh, mom and dad's pupils are not even. So we want them to be the same size. But if you have this and you shine a light in there, especially both, and the patient's not responsive, that's not a good sign. So sometimes you will hear the healthcare providers say their pupils are fixed and dilated, which again is not a good thing, because it means that they're not changing and they're big.
What's the last word?
Accommodation. And again, we'll talk about accommodation. That gets more into the physiology component.
If one eye is dilated and one is constricted, then doesn't that mean there's like that's a sign for something?
It could potentially mean that you definitely have a problem with one of your cranial nerves on one side, which could be, some sort of unilateral damage. But you would tie that into, so if you and I are having a conversation and I can have you move your eye all the way around and you can blink your eye and do all those types of things, I'd probably want to see if your mom or dad have unequal pupils. If you're unconscious, it would kind of lead more down the path that something's probably not going really well in your head, type of a thing. Any questions on that? Okay, why don't we take about a 5-minute break, because like I said, I'm going to try not to go the whole entire time because I don't want to stuff a bunch of stuff in there. So it also means that I'm not going to have really long breaks. So the component that is part of our nervous system. Everything else is again part of the eye, but it's not necessarily part of the nervous system. This is what's connecting that nervous system completely in. So we have 2 layers, and again, when you study this, you should kind of have that outline in your head. The fibrous tunic has 2 areas, the vascular tunic has 3 areas, and divisions in there, and then the sensory tunic has 2. So we have an increased amount of this pigmented layer as well. Okay? So the pigmented layer sits somewhat close from that standpoint. I'll show you kind of a little bit more increased picture from this standpoint. So here is the sclera, here's the choroid. This whole structure right here is the sensory tunic, and right down here is the pigmented layer. And it does the exact same thing. I'll show you a little bit increased picture here. This is the pigmented layer. Very, very thin. And it does the same thing that the choroid does. It absorbs excess light. It also is going to be an area where we have turnover of cellular parts. Okay, because right next to the pigmented layer is the photoreceptor. This is the neural layer. Okay, the nervous component, nervous tissue component of the eye. Now that photoreceptor, we have 2 different types. Photoreceptors are the cells that will be stimulated. They receive little packets of energies called a photon. And it's those packets of energies that strike it, that stimulate it, that then create this whole little process of vision. There are two different types of photoreceptors. They're named based off of their shape. We have cones and we have rods. So this right here is a cone, and that's a rod. There are far more rods than there are cones. Now, there's two other types of cells that I need you to be aware of. This is essentially a chain. So we have our rods and cones are attached to these cells in the middle that are referred to as bipolar cells. The bipolar cells are then attached to another type of cell called a ganglion cell, which is these cells right here. Now, you have amacrine cells and horizontal cells that are mixed in there. That has to do with processing. We're not going to get into the weeds that much on it, so you don't have to know the horizontal and the amacrine cells. You have to know the photoreceptors. You have to know the ones here, which are the bipolar cells, and you need to know the ganglion cells.
Did you say the function of the bipolar or ganglion cells is?
No. I had to think about it. Now, the processes of the ganglion cells is going to form the optic nerve. So, when you look at the structure here, this is the optic nerve. And it's all formed by the processes from the ganglion cell. Now, when we look at this picture, since we haven't gotten into physiology yet, but when you look at this picture, I told you out of the three cells, the ganglion cells, the bipolar cells, and the photoreceptors, which are the ones that actually absorb or are stimulated by a light?
Rods and cones.
Rods and cones, the photoreceptors. So light comes in this way. So why aren't the bipolar cells and the ganglion cells activated as light comes through?
Because the light reflects.
Doesn't reflect because it's coming directly through. Go back to your previous, the peripheral nervous system. Why can't I take a glass of wine and taste it by putting it in my eyeball? I don't have the receptors for it. The cells there do not respond to that. So the cells here that respond to light are the photoreceptors, not the ganglion cells and not the bipolar cells. So just a kind of a glimpse into the physiology is light's going to come through, it's going to activate these, and the impulse is going to be driven this way. Okay? Now, we will get into this a little bit more when we talk about them, but there is a difference also between the cones and the rods. The cones are not as sensitive to light because they are active when there is a bunch of light. So they don't need to be sensitive to light. And they're for bright colors, sharp images. Okay? If I'm going to a museum and I'm going to look at a painting, I don't walk straight around and look at the painting through my peripheral vision. Because you don't have sharp edges, you don't have bright colors. Because it's not hitting where we have a majority of our cones. The rods are very, very sensitive to light. Why? Because they're active in dim light. Therefore, they need to be able to get any light source that's there. Now, if we were to look at the eyeball, so if we take an ophthalmoscope, and which is the little instrument that your healthcare provider is going to turn on, they're going to come in from the side. And essentially, they're going to look, and this is what they're seeing. Now, some things are not consistent in anatomy. So when we talk about the top of a structure, okay, it's generally referred to as a fundus. The highest part of the stomach is called the fundus. The highest part of the uterus, specifically, so those of you who've had kids, after you've had your kid, your nurse is going to come in and they're going to constantly monitor the height of the uterus, the fundus, to see if it's starting to descend down after you've delivered your child. Unfortunately, this is an area that's not consistent. The posterior wall of the eye is called the fundus. More than likely it was named with the development, because again, when you go into embryological development, structures have a tendency to turn and twist and turn, and so sometimes we name them too early, so to speak. So when we look at the fundus of the eye, the posterior wall of the eye, this is what we're going to be looking at. Now, we have an area right here called the optic disc. Okay? The optic disc is where we have the optic nerve and the blood vessels leave the orbit and leave the eyeball. There are no photoreceptors here. This is referred to as the blind spot. And one of the exercises we'll do in the lab is you're going to find your blind spot. Now, when we used to do this a long time ago when I was a graduate student at the University of Oregon, is we actually calculated, there's an equation to figure out how big your blind spot is based off of certain things. That tends to get into the weeds a little bit more, but what you will be able to do is you will have an object in front of you, and it's in your lab manual, and you're going to cover one eye, and you're going to basically move your head up and down, and you're going to lose track of a particular object. But then if you continue to move down farther, that object will come back. Because what it is you found your blind spot. Now, when I look in the back, and I'm looking at the wall in the back, I don't see a big giant black hole there. Because as you basically start to mature, as you start to go through development and infancy, your brain basically starts to map it in there. We know there's not a hole back there. It's just that as the light comes in and hits my eye, where it would exit out, there would be no photoreceptor. So the brain essentially maps it in. Because you learn that it really is no big giant black hole. Okay? So that's the optic disc. Now, if we start to move slightly lateral, because when you look at, if you remember the structure where you have the foramen, which is right back here, which is more towards the optic canal, the optic foramen, where you have the optic nerve exit out. Okay, so that's on the medial side. So when we go somewhat lateral, we have this area here called the macula. Now, right in the middle of the macula, we have this pit. Okay, it's called the fovea centralis. Now, there's something very specific about the fovea centralis, in that when we look at the number of rods and cones, so remember when we look at this picture here, we found that there tends to be a lot more rods than there are cones. In this area right here, in the fovea, the center of the macula, it's all cones. As you start to move away in both directions, actually all four directions, up, down, side to side, we start to have a decrease in the amount of cones and an increase in the amount of rods. And again, it goes back to what I'm saying about the trip through the museum. I'm not just going to walk through the museum and take a look at the Mona Lisa through my peripheral vision. You're going to stop, you're going to stare directly at it so that both eyes are on the one object, and all of that is being focused in this area here. So that you're getting the bright colors. You're getting the sharp. Now when I say sharp image, I mean you can see where one thing begins and one thing ends, as opposed to blurry edges, where they just kind of meld into each other. questions on that?
So the macula is just an area or
Just an area. It's somewhat lateral to the optic disc. And again, one of the other things that your healthcare provider is looking for, and they're just going to get a gross overview because again, it's not really their shtick, as opposed to when you go to an ophthalmologist, which that's exactly what they do. They do look at the vasculature and make sure that the vascular looks healthy, those types of things, as opposed to seeing any blockages, any decreases from that standpoint. Now, just since we're on the topic, you can have a detached retina where this part does kind of fillet down in this component. Probably one of the most common descriptions is almost as if somebody just rolled a curtain down. So then you have to go to your ophthalmologist, and it's not every ophthalmologist, it's usually the ones that do very specific intraocular surgeries. And they can put a bubble in here, and the bubble then raises and it kind of pushes against it so that it readheres back up to where it's supposed to be. Doesn't sound like a lot of fun, but because you have to lay in a certain position for a while, and it takes about 1 to 2 weeks for the process to go through. Now, continuing with the structure, the anatomy of the eye. This part's a little confusing simply because of the names. Okay? So, we have sections of the eyeball. Okay? And it's essentially divided into 2 segments. And the dividing line is a structure I haven't talked about yet, but it's called the lens. Okay? So anything that is behind the lens, this is the lens right here, is the posterior segment. And the posterior segment is filled with a very, very thick, bless you, it almost is like jello. And it's called the vitreous humor. The vitreous humor is essentially developed to basically childhood. What you got is what you got. You're not constantly making vitreous humor. Okay? I'm going to talk about how we add just a little bit to it when I talk about the other fluid. There can be some cellular debris back here. So remember I said that I don't really want anything that gets in the pathway of light going through the cornea, through the pupil, through the lens, back here to the sensory tunic. Okay, well how many people here have ever just kind of been staring at the wall, or you're reading a book that's, you know, predominantly white pages, and you see a little black dot. And then when you move your eye, the black dot moves, and you get just kind of crazy as you're going. Okay? Those are essentially just cellular debris that gets stuck in the vitreous humor. So as light comes through, it just kind of naturally picks up on that little chunk that's in there, and it leaves an imprint because that's an area that you're not processing, a minute area. So it's not something that if you've never seen it, or you're lucky enough not to have it, because it can drive you crazy as you're trying to follow it through. It's not like you're missing a whole huge chunk of your vision. It's a dot, okay? And you can move it around a little bit by simply moving your eye, because there is movement in the vitreous humor. Now, anything that is anterior to the lens is called the anterior segment. Now, the anterior segment has 2 divisions to it. Okay? This is where it gets confusing. That which is in front of the lens, but behind the pupil, is referred to as the posterior chamber of the anterior segment. Anything that is in front of the pupil or the iris is considered the anterior chamber of the anterior segment. So remember, the 2 big divisions, segments. The lens is the dividing line. Anything that's going to be anterior to the lens, and that anterior segment is then going to be further divided. That which is in front of the lens but behind the iris and the pupil is going to be the posterior chamber of the anterior segment, and that which is in front of the iris is going to be the anterior chamber of the anterior segment. And again, keep saying it, and I keep saying it's confusing, so you probably should put a big asterisk there going, this is where students screw up. I should probably pay attention to the naming of it. Because that's what happens. Now, the anterior segment is filled with aqueous humor. Aqueous humor is very similar to water. And we produce this all day long. It comes from blood perfusing through the ciliary process of the ciliary body. So remember how we got cerebrospinal fluid? We had the choroid plexus, and as the capillaries came through in the choroid plexus, the plasma diffused out, it went through the epidermal cells, and at the end, presto changeo, we ended up with cerebrospinal fluid. The same thing happens here. As the blood perfuses through the ciliary process, presto changeo, we have aqueous humor. Now remember I said that the vitreous humor was pretty much what you got is what you got? There is a slight amount of aqueous humor that does go back into the posterior segment. Not a bunch, just a small amount. Now, once we have production of the aqueous humor, it's going to fill up my posterior chamber. It's going to fill up my anterior chamber. And since I'm constantly producing this all day long, what do I need to make sure occurs?
Somewhere.
That it goes somewhere, that I'm constantly reabsorbing it. So down here at the bottom, we have a thing called the scleral venous sinus. It's essentially where you have the sclerum and the cornea come together. Its other name, I think, it sounds a lot cooler. It's called the canal of Schlemm. Just Schlemm just sounds like a cool little name. And remember when we talk about the vascular system and we use the term sinus, we're talking about basically just an engorged vein. A vein that's a lot bigger. So the aqueous humor will then drain back into the venous system, goes back into the blood, and just becomes part of our plasma again. So we don't really, if I have time and I talk about pathophysiology here, this is essentially one of the problems with glaucoma where you have an intraocular pressure. So you can have narrow angle or open angle, but it has to do with this angle here, whether it's completely allowing for there to be a decrease in the amount of drainage of the aqueous humor. Okay? Questions on that? So that takes us to the very last part, which is the lens. Probably one of the more important parts, because the lens is going to help with the bending of light. It's called refraction. The lens is made up of, it's considered to be biconvex